Hello students, welcome to EPG Pathshala. I am Dr. Zinat Iqbal from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard. Today we are going to discuss about a module wherein we will focus upon the factors affecting fabrication of controlled drug delivery systems, which comes under the paper CDDS Part 1. So students, let us visualize what we are going to learn from this module. The first important thing which we will focus is the rationale of controlled drug delivery systems. Secondly, we will try to elucidate the various factors which will influence the design and fabrication of the controlled release products. If we talk of the factors, the primary things which we will focus on would be the biopharmaceutical characteristics of drug, pharmacodynamics characteristics of the drug, and pharmacokinetic characteristics of the drug. These primarily would be the three important parameters based on which the various controlled release products can be formulated. Students, before we start getting into understanding of the factors which influence the design formulation of controlled drug delivery systems, let us try to understand a few aspects about it. When we talk of the controlled drug delivery systems, you must have been hearing words like sustained release drug delivery systems as well. For the last few decades, or maybe more than that, there has always been a confusion between these two words of sustained and controlled drug delivery systems. When we talk of the sustained drug delivery systems, it primarily restricts itself to the presence of the drug for a prolonged or a sustained duration of time. Whereas when we use the word controlled drug delivery systems, we are having the facts such as reproducibility, such as the time component, and the availability of a drug in a flat or a plateau form for much longer durations. But in normal conditions or in general understanding, we usually have an interchangeable use of controlled and sustained drug delivery. To primarily focus, the controlled drug delivery system is not limited to just the oral drug delivery. We can probably have something targeted. We can have something like intrauterine. We can have something for the parenteral drug delivery, and each having the component of the time and reproducibility and making the drug available for a prolonged duration of time. In order to make such challenging drug delivery systems, the most important factors usually revolves around the properties of the drug which is being chosen, followed by the various other ingredients which probably will make a matrix or any other design protocol. In order to understand the basic premise of the controlled drug delivery systems, in this presentation, we would try to highlight the various factors which are primarily focused on the behavior of the drug. Drug delivery per se is referred to formulations, approaches, technologies, mechanisms, and systems for transporting a pharmaceutically active compound in the body as needed to safely attain the desired therapeutic effect. But due to problem of unpredictable fluctuations in plasma drug concentrations within the body tissues or organs in case of conventional dosage forms, novel drug delivery systems have introduced with new approach, techniques and mechanisms. The NDDS approach modified the drug release kinetics, absorption, distribution, metabolism and elimination with an advantage of improving drug efficacy and safety and increased patient convenience and compliance. The rationale behind formulating the controlled drug delivery system is to optimize biopharmaceutics, pharmacokinetics, and pharmacodynamics of the active pharmaceutical ingredient by reducing the severe side effects or toxicity and improving the efficacy in the shortest possible time with low dose administered through the most suitable route. The primary aim of the oral controlled drug delivery system is primarily to achieve enhanced bioavailability and release of drug from the system, which is predictable and reproducible. The design of the controlled drug delivery systems is usually based on certain goals. Before we get into the nuances of the drug delivery system design, a formulator necessarily should have certain objectives or goals in his mind. The major objectives on which the controlled drug delivery systems are designed are: number one, they should be designed in a manner that they are able to maintain uniform and predictable plasma drug concentrations. The second important goal or the objective for the controlled drug delivery system is reduction in the severe adverse effects. Of course, the toxicity should not be present there once I am making a controlled drug delivery system. Then I have to make sure that it is highly relevant to the patients. The most important thing which I can do in case of controlled drug delivery system is I improve the treatment of the patient or the therapy of the patient in the shortest period of time. Lower the dose of the drug and dosing frequencies. By lowering the dose, I am reducing the drug adverse effects of the drug. By reducing the frequency of dosing, I am making the formulation more patient compliant. As we just mentioned, that the controlled drug delivery systems are not only limited to the oral route. Therefore, whenever I have to design a controlled drug delivery system, a major objective is to select the most suitable route. Dwelling further on the rationality of the controlled drug delivery system, we need to understand the selection criteria for the controlled drug delivery system. Rather than telling what drugs are suitable, let us concentrate on what drugs are not suitable for the controlled drug delivery system. The drugs which are not effectively absorbed in the lower intestine, drugs which have got primarily shorter biological half-life to the tune of less than 1 hour, drugs having long biological half-lives more than 12 hours, the drugs which require large doses, having low therapeutic index, undergoing extensive first-pass metabolism, and having a poor, very poor solubility and or active absorption. The schema in front of your screens is primarily just suggesting that what are the rate limiting steps in conventional and controlled drug delivery system. In case of a conventional drug delivery system, the drug is present in a dosage form. It immediately releases at the absorption site, which probably is your GI tract, finally traverses into the body, and the limiting step is at the point after absorption. While in the case of the controlled drug delivery system, the whole focus is in designing of the dosage form, and the rate limiting step of the controlled drug delivery system is based on how it behaves and in what manner it is released from the dosage forms. Biopharmaceutical characteristics of the drug. The major component under this is the molecular weight and diffusivity of the drug. The drug has to undergo diffusion through many biological membrane to exert its effect. In case of a controlled drug delivery system, a drug has to diffuse through a rate limiting or a controlling matrix or membrane. If probably we have a matrix type of system, the drug primarily it has to go through its thickness and diffuse out in order to be available in the bloodstream. The property of a drug to diffuse through membranes is called its diffusivity, which is a function of its molecular weight or size. The shape and size of the pore, cavity, as well as the molecular size of the drug affect the value of diffusivity. Drugs with high molecular weight show slow release of medicament in controlled drug delivery systems, whereas diffusion is a rate limiting step. Aqueous solubility and the pKa of the drug. Drugs with very low aqueous solubility, that is less than 0.01 milligrams per milliliters are not suitable for controlled drug delivery system. The lower limit of solubility is 0.1 milligrams per ml for formulating a drug into a controlled drug delivery system. Slightly soluble drugs are also poor options for diffusion control system, since the driving force is the drug's concentration in solution. Drugs should exist largely in unionized form for better absorption, and such drugs are good candidates for controlled drug delivery system. The pKa range for basic and acidic drugs whose ionization is pH sensitive lies between 7 to 11 and 3 to 7.5 respectively. Stability of the drug or drug stability. Most oral controlled drug delivery systems are intended to release the drug throughout the length of gastrointestinal tract. Therefore, the drugs which are unstable or degrade in the intestinal environment might be challenging to formulate as controlled drug delivery systems. Oral drugs can undergo acidic or basic hydrolysis and enzymatic degradation. Drugs which are unstable in acidic pH, that is in stomach, include drugs like rifampicin, erythromycin, riboflavin, rabeprazole, mesalazine, etc. And drugs which are unstable in alkaline environments might include ranitidine, captopril, etc. Solid state is considered as an ideal state which is more stable than the liquid or the semi-solid formulations. Partition coefficient. The drug with extreme partition coefficient, that is either very high and or very low, are usually not considered to be good for formulating controlled drug delivery system. These types of drug candidates will show either low flux into the tissues or accumulation of drugs in the tissues due to rapid flux respectively. Then coming to the dose size. In conventional system, if the dose of the API is large, then it is well taken that this type of drug candidate would not be suitable for a controlled drug delivery system via the oral route. The size of the single dose in the controlled release oral formulation would then be too big to administer without difficulty in swallowing. The size of the dose. The drugs which require large doses and have very short elimination half-lives to the tune of less than 2 hours are usually not considered to be good candidates for formulating controlled drug delivery systems. Students, we would now dwell upon another pool of the characteristics of the drug which are referred to as the pharmacodynamic characteristics of the drug. Under this, the first important parameter is the therapeutic range. To understand it, it is usually a difference between the toxic and the therapeutic doses. The wider the therapeutic range, they are much better primarily for the formulation of oral controlled drug delivery systems. The second aspect is the therapeutic index. Drugs with large therapeutic index are safer for controlled drug delivery systems. Drug with a very narrow therapeutic index might show unexpected release rate. If delivery system fails in the body, then dose dumping may occur, thereby producing toxicity. Then we have an important aspect referred to as the plasma concentration response relationship. Drugs with pharmacological activity independent of its concentration, for example like rezapine, is usually not suitable for the controlled drug delivery system. Then another important part or another important aspect which will have a direct impact on the behavior of the controlled drug delivery system is the disease states of the patient. The disease condition which is to be treated by a drug in controlled drug delivery system is an important pharmacodynamic factor. Better management of some of the diseases can be achieved by incorporating the drug in the controlled drug delivery system. The typical examples of such diseases are rheumatoid arthritis. In this case, The controlled release of aspirin, for example, provides the desired drug plasma levels throughout the night, hence relieving the morning stiffness of joints. Let us now dwell upon the pharmacokinetic characteristics of drug which would govern their formulation into a controlled drug delivery system. The pharmacokinetic characterization of the drugs requires the knowledge on elimination half-life of a drug, its bioavailability, total clearance, and possible first-pass effect. The primary factor is the absorption rate or the absorption. The drugs absorbed with a variable absorption rate or slow absorption rate are the poor candidates for the controlled drug delivery system. Poorly absorbed potent drugs with high aqueous solubility and drugs which show absorption by carrier-mediated transport process or absorbed from a specific area of gastrointestinal tract are the poor candidates for the controlled drug delivery system. Next is the absorption window. Some drugs, due to the pH-related solubility, are absorbed only from a specific part of GI tract. This part of GI tract is also known as the absorption window, and such candidates are not suitable for controlled drug delivery system. Let us now talk about the another important pharmacokinetic parameter referred to as metabolism. Drug metabolism is a process which results in either inactivation of active drug or conversion of inactive form of drug to active metabolite. Metabolism of drug is possible in different tissues, but the main organ responsible for metabolism is liver, where many enzyme systems are present for different types of metabolic alterations of drug molecules after drug absorption into systemic circulation. Thus, the metabolic pattern of a drug molecule depends on the choice of the route of administration. Mainly two factors which are associated with metabolism process significantly affect the design of controlled release system. Amongst them, the first factor is the nature of the drug molecule. If the drug is liver enzyme synthesis inhibitor or inducer, then it will create a problem to maintain uniform plasma concentration of drug upon chronic administration. There are certain examples to highlight this point. We have drugs which are referred to as enzyme inducers, which include quite a number of anti-epileptics, barbiturates, particularly phenobarbital, rifampin, carbamazepine, griseofulvin, phenylbutazone, troglitazone, etc. There are other examples which are metabolic enzyme inhibitors. These primarily include azithromycin, cimetidine, fluconazole, cyclosporine, metronidazole, isoniazid, valproic acid, and omeprazole. The other factor is the first-pass metabolism, also referred to as the pre-systemic metabolism and sometimes referred to as the first-pass effect. The drug can be metabolized in intestine or in other tissues or liver hepatic first-pass metabolism before reaching to the systemic circulation. In such cases, the oral dose will be large, resulting in fluctuations of the drug levels in plasma. On oral administration, for example, the drugs might undergo various types of metabolisms at different sites. Some of them are, the first is intestinal metabolism. The drugs which are susceptible to such metabolism include chlorpromazine, hydralazine, levodopa, salicylamide, tyramine, nitroglycerin, alpha-methyl-dopa, isopreterenol, progesterone, and testosterone, etc. Then we have the bronchial metabolism. The drugs which probably undergo such metabolism include the prostaglandins, nicotine, and isoprenaline. The major source of metabolism is the liver metabolism. The drugs which are probably susceptible to such mechanism of metabolism include glyceryl trinitrate, propoxyphene, amitriptyline, pentazocine, nortriptyline, imipramine, lignocaine, lidocaine, propanolol, phenacetin, labetalol, cimetidine, and pethidine, etc. My dear students, the schematic representation in front of your screens is usually giving us an idea about the journey which a drug delivery system takes when formulated into an oral controlled drug delivery system. It will first enter into the GI lumen. There is always a risk of getting exited. Then finally, when it has to traverse to the GI wall, the other metabolic challenges are present there. Primarily, the P-glycoprotein excretion can happen. From there, after traversing that particular area, it goes to the portal vein and finally enters into the liver. This liver is a hub for majority of metabolism, and metabolism can happen via different routes and different transformations, and some biliary excretion also happens. And after this whole process, the drug is then allowed or then enters into the systemic circulation. Successful controlled drug delivery systems for the drugs which are extensively metabolized might be possible if the site of metabolism, rate and extent of metabolism are known, and the rate constants should not be too large. Development of controlled drug delivery systems for such drugs can be possible if the metabolism is predictable. Distribution. We have been talking of the various aspects or various factors which probably is influencing the design of the controlled drug delivery system. The distribution of the drug is an important aspect in an overall drug elimination rate kinetics, a rate limiting step in equilibration with extracellular fluid and blood. The volume of distribution, that is designated as VD, of a drug is used to describe the degree of distribution which includes binding within the body. The VD majorly affects the designing of the controlled drug delivery system. Drugs which are considerably metabolized before absorption can show decreased bioavailability from controlled drug delivery systems. Most of the intestinal wall enzyme systems are saturable. When the drug is released at a slower rate to the intestinal region, less amount of total drug is available to the enzymatic process for a specific period, allowing more complete transformation of the drug to its metabolites. Another viable solution is designing of prodrug for formulations which are enzymatically susceptible compounds. Elimination or biological half-life. Every drug has its specific elimination rate, which is a sum of all processes involved in removal of drug from the body, like metabolic processes, urinary excretion, etc. The elimination rate of drug is defined quantitatively in terms of its biological half-life. The biological half-life of a drug plays an important role in designing of a controlled drug delivery system. Drugs with short half-lives, that is 2 to 5 hours, paracetamol, atropine, indomethacin, cimetidine are some examples, are suitable for incorporating into controlled drug delivery system because dose frequency is reduced. But drugs with shorter half-lives, that is below 2 hours, like aspirin, ampicillin, chlorothiazide, furosemide, cephalosporins, methyl dopa, heparin, naloxone, lignocaine, dobutamine, penicillin G, esmolol, etc. and longer half-lives like more than 8 hours, which include examples like amitriptyline, piroxicam, imipramine, digoxin, practolol, phenobarbital, theophylline, warfarin, diazepam, thyroxine, etc. are poor candidates for controlled drug delivery system. Safety considerations and side effects. For some drugs, the fluctuations in plasma concentrations occur due to side effects produced by the drug. In such cases, a controlled drug delivery system can reduce the side effects for that particular drug by utilizing the smaller quantity of drug over the course of therapy. The therapeutic index is used to measure the margin of safety of a drug, which is defined as TI is equal to TD50 divided by ED50, where TD50 is referred to as the median toxic dose and ED50 is the median effective dose. The drug will be safe if the therapeutic index will be large. Drugs having very small values of therapeutic indices usually are not suitable for formulation into a controlled drug delivery system because of the technological limitations to maintain the release rates. Drug having therapeutic indices value more than 10 are considered to be as safe drug moieties. Protein binding. The binding of body proteins with a drug can play an important role in the pharmacological effect regardless of the type of dosage form. If 80% of the drug binds with the plasma proteins, then there is no need to convert the conventional dosage form into a controlled drug delivery system. This can be very well determined by a long half-life of elimination for the drug. Circadian rhythm. Circadian rhythms are mental, physical, and behavioral changes that follow a 24-hour cycle, reacting primarily against darkness and light in an organism's environment. Many biological characteristics like blood pressure, liver enzyme activity, intraocular pressure, and acute myocardial insufficiency, asthma, and epileptic seizures are known to influence the circadian rhythm. Therefore, the therapeutic effect of certain drugs like amphetamines, barbiturates, carbamazepine, chlordiazepoxide, digitalis glycosides, diuretics, and ethyl alcohol display a time-dependent nature. My dear students, let us try to conclude what we have learnt in this module. The module primarily was focused on the basic understanding of the controlled drug delivery systems. What we also discussed was the various goals and objectives of the controlled drug delivery systems, which engraved or emphasized upon the reasons or rather the ambitions of a controlled drug delivery systems, that why should we be doing it, what are the various reasons we should be going in for the controlled drug delivery systems. And we also concluded from that short discussion that it is not only the oral route, but there can be a myriad or variety of routes for which we can design and develop the controlled drug delivery systems. The major stress of the module was also to understand the various factors which will govern the designing of these systems, and these factors were primarily the ones which will be rotating around or revolving around the drug candidate. The primary factors which we discussed were the biopharmaceutical characteristics of the drug, the pharmacodynamic characteristics like that therapeutic index, the window of absorption, and the other related things. And finally, we talked about the pharmacokinetic characteristics of the drug. I hope that this module would primarily help all of us to understand the various factors which should be kept in mind while making a very good controlled drug delivery system product.